que cuida esses também. Nice. Oh my God, that's great news. You're serious. Oh, really? Have a lot of fun there.
They just had nippling.
I like you see like videos of them, great snowstorms. They're outside, playing on their lawns. It's frozen.
Should I have a second plate?
Well,
Oh, we got.
There was food there.
Oh, not me being like.
You try my name.
Expecting Austria.
Yeah, we were December.
and that's like number.
Came.
Right, you don't be outside. It's not. Acting outside of the dangerous part.
Got more time with her. Yes. Oh, thank you again. Good to see them.
Little mummy in there.
Yeah,
Biology.
which definitely doesn't look. Zoe. Started doing here a couple weeks ago.
Thank you. We are quite confident about that.
Yeah, that's why there's the same conference.
I think there's a date. Yeah, it's okay. I don't know anything about it, like, so. You know, it's out of his house. Want to take care of it. My husband.
Yeah, not the whole reason.
For a spoon.
Yeah, we talked
Sorry, you are out of luck. I haven't had one. And I have. Oh, no, we bought a house. When I started my faculty. That's my house in Chilton.
Yeah.
Yeah, but this year, yeah, so everything in law, told me that.
Where are you, Joe, now?
There used to be a golf course there. There's now like a development. Ryan boats there. Yeah, we're over there
No.
in one of the, yep, very close.
Very close to you both.
Ashburn,
Ashburn
yeah,
Country
so
Club.
now it's like Ashburn Meadows community. It's not fully done. I mean, all the houses are done, but. They actually didn't build a house, pull that built it. Sold it, they were retired and. They wanted to be in a different location closer to.
Yeah.
We're on you second, pretty close.
Oh, very close.
It's like 5 minute walk.
Between Coventry and Oak Lane.
Okay, yeah, like.
You second.
Suicide.
Nothing
fresh, you guys might spend like my age.
Yeah. Where is he?
I think they've had like some great training and.
Okay, maybe like.
Oh, no, we're out. Plus we have the senior.
Oh, yeah.
Medicare, so.
Yeah, usually I take the train to work. I drove. 70,000.
You'll probably, we do, well, maybe Elkins is a little bit closer to you. Just by a little bit. There's not too many trains. So,
You two and Sam are walking out.
Usually.
Girl driving down.
Where is it?
It is decent. Yeah.
It's not too far, not too bigger.
But I'm kind of like.
I've actually been going. You're gonna bump the rap music.
No, I think he will.
I think my transfer during here. That, like. Value.
You're talking about something.
Depending on like
streets.
who's like your team.
Did they did it?
But it was like, or my friend, the option of playing like.
It was referencing the song, and I don't remember what line he quoted, it wasn't like,
Two
but
thousand
I was like,
or something.
now all I can think about is him and the girl, like. singing this song.
Yeah, I'm not gonna watch that.
Left cheek, right cheek. But it was like,
Really.
by us. My mom, my director here at Jefferson.
No, I don't know.
This Lance, I like that.
Actually, I think we did look this up and Ellie figured it out.
How's it going?
Oh, that's what I was doing.
Did again, so.
Already.
Do I know that?
I didn't like that. Yeah.
Oh, yeah.
It's on its little headphones. And then so like you'll see. There's actually some good laser. That's a little easier.
Very good. Cool.
Now it's so much less like over.
Office next to him. We're too loud. It's now headphones.
Like three.
That's definitely for business.
I love the
Business.
Lauren's laugh, it's just so.
You knew what the, like, literally,
Yeah,
at
it's like-
Nini's defense, I was like,
You know,
oh,
scramble around trying to.
or now.
Oh.
Yeah.
It's like, it sounds like.
Oh, yeah, definitely. Come to, last year, ASM. Ironically, was hosted at, McGill University in Quebec, so it's all under Africa. And it's, you know, we wouldn't be here.
No,
That's great, but.
Nothing to
No, but it was not even the loudness, it was like, that's okay. Yeah, but you should enjoy Italy, don't worry about it. I don't know. It's I felt like it's just get perpetuated.
That's how it was with Lynn sometimes, though. Like you just get like stuck. Yeah.
You get stuck.
Like I don't know.
Oh,
Like, yeah, 2 hours, I would get stuck.
Yeah.
Yeah, we're doing that like for hors d'oeuvres.
Yeah, that was
I feel
good.
like now, people like have I think it.
There's 3 companies. This year we got maybe for Jeopardy, like children.
Now American. So that. That's just all where micro is.
I think it's people want.
Actually, maybe there's just 2,
East Coast.
and we just decided like last year, people just really seem to wanna chat and catch up and do all this stuff, so we just let everybody do that with all the
Yeah.
food around and.
It's just.
Yeah.
Yeah, I have to.
Feeling, what to be prepared for hosting the meeting this year.
Why?
Everything's, I don't know, just it seems so, yeah.
But you guys don't have like all the calls.
A little opposite.
Yeah.
Okay, I could even that's. Yeah.
Next year.
I feel like I'm always yapping in there. Sometimes I'm like, oh my god, I have to let Sharada go. See, that's why. But maybe not, she hasn't said anything, so.
Yeah, that's why we have 2 rooms.
Yeah.
I need another thing to.
Yeah.
Sure.
Yeah.
Are they those other people chat? Are you?
I think I'm rotating off. But I think Allison and Ken are staying.
Me to go to the induction.
Really sure.
I never.
And I don't know what their plan is. Last time they just asked me, so I don't know if like
It was.
the executive council already has people in mind or what they want to do.
Yeah, now we.
Yeah.
You don't get any, and then it's like, okay, well, if I'm.
Much better than the opening.
Yeah, I know, I don't like that. You like the 2 drink tickets and
Yeah.
then.
I could get all.
Yeah, congrats to Mr. Dowd is like a collector.
Yeah,
I
I distract him so bad.
No, or like you would distract. I feel like it's like
Yeah.
speak up and then I'm not around and listen.
Yeah, the opportunity cost of like sitting in that line for a free year versus.
I've been there, but that's.
Going for the whole time?
No, I don't know. The first year.
Roland's coming with you?
Yeah, unless they're in our church club.
Yeah.
We're like. So.
Thanks.
Yeah, we should maybe like.
Well, we just.
Well, I think.
Not so. It's not.
Yeah. Use that.
Like, we don't have any.
Yeah, like the. Like a dinosaur on the team, like certain.
Like that.
It's the only vendor that I went to.
Yeah. I get so, yeah.
Three board thing that's scheduled on Wednesday.
I just asked. I'm on like.
Yeah, I don't think that's like.
Just.
Yeah.
Yeah.
Oh, that's what I did last year for like months.
But it's also like biochem.
Yeah, I was gonna go on Thursday to go like.
Yeah.
Yeah.
So I do.
Oh, you're gonna go to that?
I was gonna, since I'm a representative, shouldn't I go?
I went once.
Week or something, like, I don't know.
Well, I think I actually know it. But I think the older people.
All the opening sessions to go to. I literally just walk up on the stage, take my award. It's not my talk or anything.
Yeah, like, come
Go.
for, I didn't come for that. Last year I sat, that's the first time I ever sat.
Oh, yeah.
No, I.
That I will have is the only flag number ever from her.
I went like the first time I've ever. So. Opening session since. But I did go to the range nights. Makes you realize like how. And how well we're doing.
I know, I was like, wow, we're doing.
We got into these small groups and we were talking about like. Yeah, we were talking about like how to, you know, what we were trying to do to improve our things, whatever, and this one group was like, how do you have money to have? Well, which size you got? Well, it's like you can't find a way to get $1,000 from cobbled together from, you don't think a big place. I don't. But I was like,
Bacteria centric
you
and.
can't just send some blast emails to like 30 companies in the space you know and ask for $100, like.
That's how confusing.
You kind of have to get it off the ground, like, not like.
Yeah, I remember one of our meetings actually. Meetings, and I was like, yeah, it's like how we fund our. You know,
Yeah.
yeah, I remember if you're in the scholar educational branch, this stuff, or maybe this is probably right off on the line this year, after. Like a lab. They only had a couple meetings a year.
Yeah, I was in.
Yeah.
For
A couple of them would have big.
Like, oh, no,
Like, maybe just like to have like one
It's like
year
this
now.
stuff.
Like, how much money do you need to do that?
Yeah. Notice that like the
Yeah, hard to
box changed after our like.
Yeah.
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 It was like in the head, but like everywhere, like I think.
Years
later. Oh, my job.
Like the other year was like.
It's.
Yes.
Yeah. Like I feel like. I bet not.
Yeah.
So that's September.
Yes. Allison texted me right after.
Yes.
That's.
Maybe so.
Soon forgot.
I'm a result client.
You have little.
First day job, then I have my night shift, so being a driver.
I remember those tests.
You guys.
Yeah,
Yeah.
you know, and I knew I was trying to.
Yeah, right. Yeah. She does. Oh my gosh.
That was no good.
Run over and grab this.
Shirley and I are like,
Oh, no, that's fine. Yeah. Uh-huh.
I can always run over.
TK on that.
I'm sure that's done most of it. But like, there was.
I mean, they can look at
Weird, like.
I'm bringing my car.
That's so funny. I mean, like, it's word.
Yeah.
I mean, it's not. One second, just, yeah.
Research on microbiology.
In my last, like, my office area, so.
Privilege of giving another talk. Micro.
Yeah.
They were kind enough to schedule it right after the session I do.
Yeah, so there were just like.
I have a tote bags in my.
But there was a story.
Yeah. Maybe before that, and I also like have like for the like chair. They did it there. It's like. They were like the.
Yeah.
Most of the battle of the breeds.
They're nice. I'm like.
And now the quiz.
Pretty.
It's always the first thing on the first day, which is really nice. But even though I know all the answers, it's like the most draining session I've ever done. It's 2 hours, you're like, have to like keep it going.
Quiet participation.
Yeah, you gotta get the audience, the people. We were like 2 hours, so I usually like go back to sleep. Get back, and then I reemerge after lunch. That's fine. And they were like, yeah, your work talk is right after battle of the breed. So it's like, fine, I have like 3 more days, can't you put?
And yeah, it's more like.
I'm gonna sleep up there.
I didn't know you gave a talk.
Right.
Is it like? Is it in the ASM?
Really put us in, myself and,
like.
Katie Mathers. And they put us in a public health position. That's interesting. But they told us we could talk about whatever we want. I was like, well, I feel like I have to tailor it to.
Public health people, yeah, because are there other public health speakers? You guys are the 2 clinical.
Yeah, there's like 1 speaker
Isn't it
and 2
just
award,
health?
there's 3 award recipients, like some of them make sense, like public health, and then 2 or 3 from the. So on Thursday, I have a industry like.
Never say yes when there's something or something.
And then Friday morning, I have to roll the brains. We followed I have work on. And then on Saturday, I'm doing one of those ASM studio case things for the book.
It's not my problem.
Go first.
Just comment.
Yeah.
Just kind of.
That's it. I can't even get the award winner for your registration.
Whatever happened with the Peggy Carter?
They did adjust medical lab scientist.
We don't have any feeling about it, but it's fine.
That's all you can say.
It's cheap though. It's like $300 as long as you're like on time. They did, I pitched this like whole new session on. I think it's on Sunday.
I think I've listed that. I wanna send it to our lab.
Have a market.
I don't even know when.
Haven't announced it yet.
Right, I missed. It's not.
So they've been, ASM has been promoting that there's like no medical lab scientists. Attendance to microbe anymore, which a lot of places have removed.
Yeah.
So I said,
So.
We'll make like a MLM centric day. We'll have like this jam-packed CE, they get a bunch of credit out of there. Give them lunch. Bring them to the vendor floor so they can meet people and see some focus for a little bit of time, etc. They were all about it. We actually like built up this entire 1 day crash course. So it's on a weekend, so we don't take a day off from work. It's like, make it free. Which did not originally fly. But I was like, I gave him like the ringer at one, I was like, say you want this, and like, you're gonna charge people that, like, you either want it to try and bring it on the road. 3 or 4 years, get it to be like a really well-known play, then when you have more MLS participation, then
Amazon student method.
they are, they're almost like, we don't charge people anything, they'll just sign up and then like not actually come. So they, I think they're $25 or something. But
That makes sense, actually.
yeah,
Sure.
but it's a 1 day, it's totally, from here, you take the train down, you go to your 1 day thing, take the train back, like.
Yeah. Because I was gonna send it to the people in our lab, because there's. Around schools, even like their supervisors who aren't gonna go to the actual.
They probably get more out of the vendor stuff than like any random, me as a random grad student walking around, like, I had no idea, but most of those vendors are clinical stuff.
They're just so bad with like, connecting the dots. It was like, have it sponsored. Whatever it costs to cover the food and the distribution. And explain it to the students. These are all clusters, not currently members, which means they're not going to, so they're like untapped. Audience. 1 day, yeah, 10% of them will be a supervisor in the lab. And they will be your target person, like invest in the thing. And I think like the vendors were all into it. They're gonna cover the food, I guess. But it's just like,
It's just easy, like.
yeah, they outlined a whole day and it's like, you know, basics of AST, like mechanism review. Lars Westblade, all the diagnostics and challenging cases, Sean Zag.
Oh, nice.
So like,
Yeah,
they're not like,
Yeah.
right
Yeah,
people
great.
to do these things.
Yeah.
But no, for like the defense.
See if it works. Because they don't start announcing it soon.
I know about it because of yes, like for example, and because Nicole emailed Matt asking if there was like someone from our lab. Example, something, and that's the only reason I know about it.
Nicole, that Dermot?
Nicole Jackson.
Oh, from ASM.
Yeah. But they need to announce it.
You're trying to convince people, yeah, that now you should.
Literally in
Right.
Yeah, they get from this weekend
There's-
because like some things are really
Should.
strictly.
They have the whole thing. They were like, we're working with marketing right now.
No, Machino's finished. This is her 6th year too, I guess. 6 or 7. I think it's her.
The thing I think that like DC is a perfect place. There's a lot of laboratory systems around, and then there's even places that aren't all that close. But you can easily get to DC. So like all the Baltimore labs are within reach. All of the Philly labs are within reach. I would say like anything in Richmond and like the Virginia area is within reach. So it's the perfect place to like kick this thing off because there's a lot of people that would just come in for the day. And then depending on where it is next, like.
So she's. That's they said where it is.
During the.
No, but it's in,
Like, wow, this is.
Not publicly anyway. I was trying to find it.
I know where it is. I know where it is.
No, I don't know where it is.
She's in York.
Yeah, so I get so sweaty walking to my hotel. That was bad. That was so bad.
It's in Chicago.
I like Chicago. Chicago over summer.
Like later.
They just now have.
Yeah, it's good.
Yeah.
Performance Center, wow.
Like Greenfield.
Yeah.
They do EDLM.
How longer was that?
Here, I'll show you, I have her.
Definitely.
Was it before you guys created it?
Ryan Relich and Aaron McElvania.
Oh, cool.
Kempe.
Did you say that they're in?
He was a hyphenated name. And before them, it was Chris Dorn and Susan.
Looking into Trader
And I don't know, originally it was Peter Gilligan.
Joe's.
Oh, I just.
Haven't seen in person for.
Well, I was away.
But I don't know, so I don't know if it was Peter and Roberta and Susan and Chris, or if there was like a set of people in between.
Next day, I can't.
Yeah, that was really awesome. So.
Still need two people. Just
12 days.
On the team, right? Oh my gosh.
it's anyone who's involved in CPHM subcommittees.
Yeah.
Know that, I know.
Right, a bird.
And done it once, and I was so nervous every time. Yeah, we got second, we all.
Yeah.
Like, please. But I'm just my first now because sometimes it's me and Venus and check it out.
The red hot, really, yeah, I remember that. That was my first year doing it.
But that's.
I didn't host it, never mind. I think I was taking notes because I knew I was gonna host it.
And say, so I remember him asking, okay, so
You're probably gonna need at least one.
I was in Houston.
I get really nervous.
right at she very like, do a lot.
Yeah, but I'll
Not because good now, I can tell you that.
tell everybody that fellows are at a disadvantage because their brain is full of like actual, or it's practical things, and none of those are on.
Nice to see you.
If you need someone.
Can I tell you, like, I'll give it a little more time.
Who's on the team?
Think I'm on another.
Ortiz, who's like vice chair of CPEP,
Honestly know about.
I haven't.
and the last person who's on
Like they're affected.
whatever, Sami,
Yeah, but I do.
Yeah.
continuing education.
I got through and now they're.
Yeah. Now, she calls it like CMS.
You went to another line. It's the same.
Sami is the chair.
You know, like Southampton chairs, I don't know, yeah.
Day and a day before.
They had
So like.
really same day.
Professional development, work and activity, it's like pretty similar.
That ship was.
But I'm on the. all that one.
You're on Rich's one.
Yeah.
Yeah. I do.
No, they'll like that. You're just like.
Oh, yeah. No, Sami
I recall.
is another. They're the ones that are supposed to be putting together like the YouTube videos and stuff. She's always talking about.
It's
Videos.
If anyone has. Yeah. If you're. Yeah.
My son doesn't.
Yeah. Two on Thursday and I have not started.
You would have to explain, multivariate regression.
That is my problem.
I don't know what I'm looking for.
You get a lot of variables and trying to find what on.
I'm gonna go to, so she has both back years. Yeah.
We still alive.
Thank you, Sandra.
Yeah. You did? Yeah. She gave it.
Like random. Lab is good.
Well, yeah, I haven't talked to you.
Said the same thing.
Let's make sure you at seminars.
St. Jude. Very far along, St. Jude.
I think we'll be.
Hard to get away.
So I think she wants to go back to Texas really bad. But, you know.
I do think she's interested in having a job.
Yeah, job's a job. And it's closer.
It's just like.
And Memphis is a huge area.
They would drive the 9 hours to her family. For
Oh.
dogs. I guess. Fly with the big dog.
Well, yeah, they're both, I guess. Otherwise, you can do a weekend here from Memphis.
yeah.
But at the airport,
Very for this trip.
you can fly this car.
Tuition, I feel like you can't really ask me about.
Is there more than one direct?
There used to be.
She didn't ask that. I wanted her to be in the Austin, but I don't know what Heather.
I don't know either.
I think that she is leaving, and that is the open.
Here's the-
Isn't that?
I don't know what Heather Glasgow's doing, because Heather Glasgow LinkedIn friended me or whatever the other day, and she still has her job listed as St. Jude.
True.
So I don't know, but it's supposed to be replacing Heather.
Yes, Randall hated.
Yeah, Randy Hayden.
He's been there for that 30 years or something.
He's like 105.
There's still like checks that are.
He's like super virologist. He's like an original EACV member.
Yeah,
he's like
very
a
distinct,
short-
like very distinct.
Short guy, goatee, gray hair.
She like crash something. Yeah, short goatee.
Oh, yeah. Yeah,
It's other,
like a real
like a
kind of-
microbiology, yeah.
We're not.
Had to go through, yeah.
And but it's funny, he's the division.
So March
The division has four other microbiologists and then like EPAASM person. And then
Okay, so I think we're going there.
it's because they put like self
Have
with
the
the
England and we leave from there.
And then after.
I only know two persons that have jobs. Yes. I don't know.
So
It's
excited that
it's the fellow from Vanderbilt. She wanted like person,
Oh, no, wait.
like fantastic AST stuff, so she got hired out.
Oh, that makes sense. Buddy.
Before it stops.
Yeah. Her name is.
No, it's even.
Denise or Denise. Yeah.
That's what I.
The job I'm most curious about is the marrow. Because all Nikki left. Well, Nikki left like
A year ago.
A year
They
ago.
had culture spot.
they
Closed
closed
at
that.
Cincinnati Children.
Remember Cincinnati Children's did a job opening like 9 months ago, whatever.
I like all the musical chairs and athletes.
So Nikki Putnam's job like got created out of thin air, apparently, when she finished it. And then she left to go to Pattern. And they left. And then Paul left for Cincinnati Children's.
Just.
Yeah.
Lunch is nice.
Doesn't say where. Yeah.
Because I was talking information, and we could tell them.
I feel bad for Lou.
Yeah, like, cause you
He just moved like this week, last week.
I know that he was trying to get back.
Yeah. Yeah. Last week was moving day. Wow, Wednesday he left.
There's the two.
You guys watch the ASM?
Essentially,
Robert.
I had French.
aspect. Think about
How much does it cost?
It's like an AST.
About AST size.
Oh, was it DLSI up there?
No, I don't know about, it's always. But anyway. How do you ever watch? And I could easily. It was nobody is.
How was it? Yeah.
Needs to be on title. Fresh and broccoli. Just use. Fresh broccoli. Oh, Robert Bernard.
That's the whole.
She's the head of the CLSO. Audrey?
No.
Today.
Amy Mather? Yeah, that.
It's
I realized that, because you didn't say anything. You said
That's Trish.
I'm the one who moved it.
is open.
And she also said.
As soon as I said, because the CLSI was open for the UCAS.
Well, it is in the sense of any part.
Instead of 5 per minute, it's like 6 per minute.
So that's
Right.
Can't listen to the
Round trip.
the dictatorship of fussing.
Oh, yeah, that's funny. Then UCAS says, here's all of our stuff. CLSI is like, no, give us people first.
Well, that's true. Now they cost.
there are ways.
Yeah.
Idea.
Yeah.
Well, how do you go around it?
Oh, that's interesting. I know that. Oh, that's cool.
Each side.
Yeah, but it is kind of different, isn't it?
It is. Thank you.
That's not hard.
No, no, I know. If you go to.
Webinar.
Yeah, they were.
It was submitted.
Hi. Well, they had someone.
I'm gonna like, it's gonna make one for everybody in the lab, so I just like was handing this out. Yeah. 3 times this is gone to.
Did I just?
Yeah, the, right.
Standard.
You do, but it's done. Yeah, I told the doctor and say somewhere doing it 1 year.
Just.
Really don't tell you, it's this silly about something.
But I remember how to do it. Yeah.
Yeah, I was like, Robert, let's say all this kind of free.
Yeah, right.
So like.
The Cleveland, Western. Yeah.
If you have gauge, you have to say,
I like him and cheaper, like.
yeah, oh God,
Yeah,
yeah.
I bet.
I mean, he's younger. Well.
Yeah.
The paper that was the title was like like in-depth analysis of shit or something like that.
Remember that. He's
You're doing it?
super smart. He can explain things that anybody. Well, at least he'll decide and I guess conversation of rabbits. They talk a lot.
Like, oh, what's that?
Back to the advisor. I'm like talking it back. It's like, not just like.
You're in the, you're in.
That's the type of thing, yeah.
But then the track that. To stop.
See what I'm saying? Well, fun, it's like, it's all
Like, they're bad at it. I'm like, they do like, that was about it, a straight line.
Right, so then you just say. Let the physician make.
But I know some.
There's a lot of people that want to the chair for. See what happens.
I do get to encourage you.
Try to avoid viruses. Let's find that.
Let me make proposals. I don't think.
Not that like.
It's just like. It's nice.
Elements office, I've heard of it, I work with AHM.
He said, it's sunny next guy.
Oh, okay. So instead of talking like.
Yeah. Well, I like that. No updates. Stay the same.
I want a good graph.
Wow, every third Wednesday.
Oh, so you got a little thing.
Well,
You gotta.
you keep going, and the thing of it is.
It was right.
Yeah,
But now that I'm just editing.
And then you're just like renewal. Okay. Let's talk to Sunny.
For next year.
So
Maybe something like this, Patty, then.
one had it in last night, 7 for the October. It's ours. Yeah, he might need a higher history. And I'm like, But I was when you're
What do they need the? For?
After.
you're like, I don't have a list of attendees, I can provide you a list of every single member.
That's helpful.
I mean, it's like, there's literally like 500 people on it, and it's kind of like a problem.
The question I was asked was, are you going to a meeting? And I said,
After that.
The rest of this. Like, you know, we have to like. They've never asked us.
Sent you a list of like every person that's ever been.
Yeah, it's so funny.
Like,
You're gonna learn
just export.
more data, not good.
Yeah.
Yeah.
So do you?
So that work was really easy.
Whereas like.
You could go into a behind the scenes mailing system and website and literally just click the lists you want and export them. It took me like 10 seconds.
Well, if you were like, I don't have this, I was. Oh, no. Our executive dinner, the day, these people will be there. And I was like, that's close.
Closer to 30 than 500.
They're in front of you.
So then.
Slides in today? Not going to these slides.
No.
Said that's an option, right?
You can do them.
It's monologue.
That's because.
I think,
Yeah.
it's two slides.
Right.
Yes.
Yeah. There's no rules. People just have historically gone with slides.
Really?
But I think you'll lose people.
Well, it's because I couldn't decide. I mean, there's too many things to choose, and I didn't think of one thing would have adequate. Right, my.
You're the one who told me about it to begin with, yeah.
Spiel could have laid out.
Strategy.
Yeah.
Very small, very experienced.
Yeah.
Not very. It's getting with the other teams, I'm trying.
I hope it's the sign you want it. By showing up on time for the meeting, you got.
It's just what I wanted. Yeah. Well, it's funny that then like.
So you mean like.
Getting in early and then Esther emailed.
Yeah, I'm ready now.
My predecessor.
I actually hopped back into the meeting and some random phone number was on there that was just like,
It's like.
it wasn't Jess, but it was like just someone talking. And I was like, this is very weird.
I set up a rush here. Vaccines like.
I think David, is there.
Don't worry. I got like.
Yeah.
He shot his fellow rock.
Wish we got. Rush to the.
President fashion, yeah. Versus like, I'm in my third year.
It's nice that it's shorter. It used to be an hour and a half.
Yeah, I think it
We had like,
This guy.
like almost on each side, to like, make it was like a long presentation.
No, what is that?
How's it just like the side?
It's like 10 minutes, 10 minutes, 20 minutes, questions, 10 minutes, 10 minutes. Key to the debate. To get everything.
Yeah.
So, other time, at the beginning, and
His testimony.
I changed the vibe.
Yeah.
you need to go around.
Yeah.
Well, music sucks. Back my notes that.
I can see this is funny.
The 8 sequences.
Man, another.
You guys had a
Don't send a lot.
sequencing a,
Don't send a lot of it. That's.
yeah, you guys have a crazy intercopys.
Really, you know what, the email guard.
You do?
The first season. Yeah. It's not like, really to see how like.
They just like resistant to everything.
Fire like, is how like. It's available. It's like, labs is like, probably is how.
You're in charge of everything.
No.
We've had this weird juggle of getting because of spring breaks and other travel. We moved it.
How many 70 pages in the house? Someone who likes it.
Yeah. The second one.
Once you get used to two on, the third one is just like
You just maintenance that.
They used to do fun, which is insane. I
For the
was
time.
yeah, not even 4 weeks. Like,
the month of March.
that would be fine. February.
Which is not the weekends.
Yeah, I always wanted to, I think I knew.
There are some weekends where you can get like 6 or 7.
Because like spawn like.
It's just annoying, like.
Yeah. Yeah.
Here, epic.
I'm okay with the phone epic, it's when someone's like, on one of the Cerner, like that. Or like the.
Right?
Yeah.
Even if you're.
Yeah, I'm sure. It's not.
And I was getting attention. He all the like,
It's just that each one is individually.
and then optimized and that was this kill not matching the. I mean, you still.
I'm not.
No, absolutely not.
I technically not. I was just. I know technically. So I thought it was.
I just that they have had a big tablet or a big.
Okay.
Yeah.
I have actually gotten the Cerner.
Whoa.
I was so annoyed by something. You can like, the phones now, the hospital phone, have a VPN. You just like go to your settings and you VPN and you like slide bar to the VPN. It's really small. And then you can go to like the internet and then to the CP call page and then pick up the Cerner app. And then
It sounds like.
which and then you're like, when I do the accession, so I took the accession.
No way.
I got all the way to the work. Which is amazing. It is a web based. There is not.
I have to get patients.
Yeah.
But yes, I actually got it, sir. Yes. Let's try to get it all the time. It's the straight.
Well. Audiobook of that, you say, well, I didn't really understand how it was really.
Just the.
Yeah.
Oh, they'll like summer student.
I can see some busting well.
Jefferson ID and whatever.
Really?
Not media, I mean, all the lunch. Like fine, like their husband wanted to try and have like super place because the testing issue and then they had to find something like.
They still use, would they still prescribe a drug? Rest of day, that's see for the.
Like for example, just reporting place for like.
Yeah.
Like Tazam.
Yeah.
They have like a mega that one of them. Just start thinking.
It does not go for start thinking. It's now less than.
And then sometimes they'll ask.
There's only 2 people on type information versus.
Yeah, I would be nervous about that.
One or two times are like.
Yeah.
We have a lot of things that go around. Yeah, I'd be anxious to see them like suppress it.
Yeah.
Just don't show anybody.
But I like,
Really figuring that out.
if it's like ambiguous at this point, it's not like actual suppressor, it's like we're.
2 hours.
And then delete that line so they don't accidentally.
Yeah, so it's like.
It's not an official.
Gosh.
I'm in the thing right now. 7 hospitals.
We might.
I'll discuss something. Oh, this is just a information gathering session. Yeah. Want to understand why what everyone's doing and
Big special.
justification for you. So you're
The F is here.
together.
Yeah.
I get any like CMO
So there.
principals will be retaining all their current workforce related to, and I'm like, how do you even know? It's like, do you have my office booked? Like an hour ago. All I said. Could you put your information in the spreadsheet? Just wanna look. Everybody's going. Some people are cheating. Oh.
Jury calls. Yeah.
Chair pathology website calls it. But it's really to
Yes, this is the way.
a stack.
We care a lot about micro.
No, he just cares a lot about
Something along the lines
the impact of life to stay. He's strapped prenatal screening. It's done outpatient, like weeks before delivery.
What?
Like, extra stays are. Oh, yeah. I meant, they move off really fast. Stack works. Every group is.
I've been around
Like Ron.
Oh my goodness.
Things stack.
I don't know.
Yeah.
What?
He's like going down his like.
It's like ChatGPT where you keep pushing back, where like, are you sure about that? It's like, no, you're right. I'm totally making this up.
He like has a.
My job is just staying in.
Like where it's from like, he just goes down and goes every time. Hang. LJ.
In my seminar. No, maybe.
This is charity.
I think they technically. See him. I think it works. I think we got everyone works.
All right, everybody, welcome to our meeting, our monthly meeting this evening. And so I'm Jason Heindel, and I'm one of the co-organizers of the monthly meetings program for the Eastern Pennsylvania branch of ASM. And our speaker tonight, I'll introduce her in a few minutes, but I have a few slides to introduce the program that is bringing her to us. So this is the American Society for Microbiology's Distinguished Lecturer Programs. We do want to acknowledge what they do and have done, and the fact that they provide funds for this program. So they provide some slides that we have to use. And so this is, they've been around for 60 years with over 1,000+ lectures. And we do get at least one ASM DL lecturer per year, and they're always excellent, well-qualified individuals. So it is a great program. So we are appreciative of that. Also want to talk about ASM. This is a slide where ASM would like you to join ASM National, but we would actually like you to join us. We are the local branch, it's the Eastern Pennsylvania branch. If you are a student, the cost is amazingly free. So you can see over there, it's free if you're a student to join the branch. If you're not a student, if you're faculty or postdoc or something else, it's only $20 for an annual membership, which is pretty affordable. You can also join ASM National, which we would encourage you to join ASM National. The cost is not necessarily terrible, depending upon your employment status. But do visit our website and consider joining the branch and/or national. I also want to highlight that ASM, one of the things that they do on the national level, they do advocacy. And right now, this is obviously a very, maybe not obviously to some, but a very important thing that they do. So if you want to be hooked into their advocacy efforts, either to participate or just see what they're doing, I do recommend you can scan this, or you can just go to asm.org/advocacy and get involved. Sometimes that involvement is as simple as sending an email to your rep, your senator, something like that. So it's very easy to do something. And there's lots of ways to get involved. So definitely check them out. They're doing a lot of good work on your behalf as microbiologists or as scientists in general. Whoops, backwards. So for the branch, we have one more monthly meeting for this academic year. It's in May, on May 18th, and it's the student chapter organized meeting. From 4 p.m. to 7 p.m. It's our 30th anniversary of the student chapter. So it's a big deal for them, a big deal for us, and we're very excited. We would like you to come out. So stay tuned for a list of the speakers, multiple speakers, including trainee speakers will be presenting. So a really good opportunity for them to present and you to hear their work. And then we'll pick up with the monthly meetings again in September. And if you were anticipating Ralph Isberg's talk for what we call Legionella Fest, that got canceled because of snow, that's going to happen in September. So stay tuned for those details. All right. So now it's my pleasure to introduce today's speaker, and I'm going to do my best here. There's a lot of information I could give you, but I don't want to give you too much. So our speaker is Dr. Vanessa Sperandio. I hope I got that right. Sperandio. She says it's fine. She is currently the chair and a professor in the Robert Terrell Professor and Chair in the Department of Medical Microbiology and Immunology at the University of Wisconsin. And she has a whole number of accolades to her name. I'm just going to list a few of them for you. So she did her training, her bachelor's, master's, and doctoral training in Brazil, in Sao Paulo, I believe, and then came to University of Maryland for her postdoctoral work, and then is now, after a stint in Texas, is now in Madison as chair of the medical microbiology. She was a Latin American Pew Fellow, an Ellison Foundation New Scholar, a Burroughs Wellcome Fund investigator. She has been a National Academy Kavli Frontiers of Science Fellow. She's a fellow of the American Academy of Microbiology. Let's see what else I got here. 2015 ASM Eli Lilly and Company Elanco Research Award winner, GSK Discovery Fast Track Challenge Award winner, and a fellow of the American Association for the Advancement of Science. Everybody's honking for you. So shout out. And she does serve ASM National in a number of ways, in particular through the American Academy of Microbiology. So it's our pleasure to welcome Dr. Sperandio. She's going to tell you about the highs and lows of enteric infections. This out. This, let me just think. Try that one. Let me just hide these so they're not in our way. Hide.
They don't touch.
Yeah, don't touch anything. But you should.
There's a very big sign in red.
There we go.
Thank you. So first of all, thanks for the invitation to talk about our work here. And as per my title, I know it's late during the day, we are going to be talking a little bit about drugs. So my laboratory works with the enteric pathogen, primarily enterohemorrhagic E. coli EHEC 0157H7. And it's responsible for outbreaks of bloody diarrhea throughout the world. This is a foodborne pathogen. You get infected by contaminated food and water. Usually you get bloody diarrhea, which is not fun, but it's not going to kill you. But the young and the elderly can develop a complication known as hemolytic uremic syndrome, HUS, that can lead to high levels of morbidity and mortality. So these outbreaks tend to get a lot of press. This is a true pathogen. So this has a very low infectious dose, just 50 to 100 colony forming units. So it's really hard to control in food, and that's why there are outbreaks every year throughout the world. And it colonizes the large intestine of human beings. So this is kind of the scenario that EHEC is looking at when it gets inside of the host. First, it needs to see. Does this work? I'm not gonna touch. First, it needs to know that it's in the large intestine here, and then it needs to know where it is in the intestine itself, in the colon. Okay, so your colon has your epithelium here that's protected by a very dense mucus layer that's free of bacteria, and then you have the outer mucus layer facing the lumen, and this mucus layer is highly colonized with a very proficient microbiota. This is where the microbiota is at most dense in the intestine. This is also highly innervated by the enteric nervous system, and one of the neurotransmitters we're going to talk about today is norepinephrine and epinephrine. So 50 EHEC cells have to come in and find a niche to establish themselves in the intestine. So as any enteric pathogen worth its salt, EHEC has mucinases, so it can destroy the mucus layer and gain access to the epithelial lining. Once in the epithelial lining, they express a molecular syringe that we call a type 3 secretion system. And through this type 3 secretion system, it will inject about 51 effectors that will either mimic or hijack mammalian function. And what happens then is microvilli gets effaced, so you lose absorption, and EHEC also change signaling, do immune modulation, and it changes the cytoskeleton, forming this pedestal-like structure here that it uses to adhere membrane to membrane to your cells in the enterocytes, which unfortunately for us, this means we get diarrhea. So every time we talk about EHEC, this is an E. coli, and people assume that they usually think about E. coli as a commensal or a molecular genetic tool. But not E. coli are equal, and they tend to have a lot of different DNA on them as a species. In the case of EHEC, it has this pathogenicity island that I'll talk a lot about today, because it's absolutely essential for it to colonize the host and infect the host. And I'll call it the Li. It has 41 genes, and it encodes for a type 2 secretion system that's highly regulated, which are these molecular syringes. And you can see here a lot of gram-negatives use this, Shigella, Salmonella, but they have small syringes. EHEC tends to have these very big syringes. This is actually the type 2 secretion system from EHEC. And you can see this is a huge structure, so it's expensive to make. And what I tell the Salmonella people in the crowd is that EHEC has a type 3 secretion system much bigger than yours. So, and then it basically formed this pedestal, so you know what you're looking at. So this is EHEC expressing GFP. The HeLa cells here, it can do this in any epithelial cells. The cytoskeleton stained in red, and beneath each EHEC, I hope you can see there is a little stalk, a little pedestal, and it's adhered membrane to membrane in there. Okay, so EHEC has a very low infectious dose, is in the intestine, dealing with this very, very big and adapted microbiota. So in terms of species, there is a lot of different species in there, and we all have our little fingerprints as what type of microbiota that we have. But in the terms of phyla, the main phyla of a normal healthy microbiota is dominated by Bacteroides, followed by Firmicutes, and this little red part here, these are Proteobacteria, which is basically your commensal E. coli. And there is a lot of signaling that goes on in there because they have to coordinate behavior so they can coexist. So they will use things that we consider signals and also use carbon and nitrogen utilization, both as signals and as different sources, so they can coexist. Okay, so I just gave you my age right now. The young people are confused. The older ones know what I'm talking about. So one of the things that EHEC reads in intestines are sugar sources. So as it says here, EHEC sings pour some sugar on it. So this happens at multiple levels in the intestine, and since this has been published a while ago, I'm not going to go into extreme details. I'll just give you like the highlights. So Lenny Pacheco, former student in the lab, figured out that to sense the lumen part here, remember in the lumen, EHEC does not want to make the type 3 secretion system. That's not where it adheres. You saw the size of that thing, right? So, and in the colon, most of the monosaccharides and disaccharides, which is what E. coli can use directly, are absorbed in the small intestine. In the colon, the main sources of sugars are the sugars decorating the mucus, as well as complex sugars like amide or pectin. So EHEC has a friend here called Bacteroides stephathiomicron, which I'll call P-theta because the name is huge. And this is a very saccharolytic species of Bacteroides that basically uses 18% of its genome to harvest sugar sources from the mucus here, so it can use and also give it to other bacteria that are there in the colon. And the main competitor for EHEC is commensal E. coli. And one of the sugars that's really prominent here is fucols. So lots of bacteria use fucols, but EHEC has one signaling system okay, that's only present in EHEC and not in other E. coli or enteric bacteria that Aline called FUSK-R. Okay, so FUSK is in the membrane. It senses the fucose that's being released by Bacteroides, and it modulates expression of the type 3 secretion system, the lead genes, and the metabolism. So EHEC can read that it's in here in the lumen and adjust so we can compete with whoever is in there. Later on, I told you it has mucinases. The mucus layer is gone. Okay, all of those sugar sources are gone. This turns into a very gluconeogenic kind of environment. Beta then has to start making succinate, and that's read by EHEC. Like mucus is gone, I'm in the epithelium. It's time to use this to really express the type 3 secretion system. Okay, so it doesn't stop just with what's in there. Angel Jimenez in the lab has shown that one of the things that's also sensed is this dietary sugar called galacturonate. So galacturonate pectin, okay, is made of galacturonate. And again here, beta breaks pectin into galacturonate, so EHEC can also use that as a signal to modulate virulence, expression, and metabolism. And because if you put people in the lab and give them a little bit of freedom, they don't stop. So Reed Pfeiffer and Zelia Menezes have also shown that EHEC is reading amino acids, fatty acid sorts, and there is like at least 72 transcription factors in the end of the day that are regulating expression of that monster of that type 3 secretion system, responding to different cues. Okay, so enteric pathogens exploit the microbiota, which usually we think as a protection barrier for enteric infections. But there are bad actors like that, such as EHEC, who knew how to explore that. And that brings to the point of like maybe different intestinal microbiota will change your susceptibility to gastrointestinal disease. And the microbiota changes, as we know, according to diet, environmental insults, such as antibiotic treatments, and you also have genetic components that change the composition. And we wonder what happens when you mess with the microbiota and if we can manipulate that to treat different disease states. So Fernando Martins then decided to try to look at interactions between the microbiota and EHEC and the host in a more mechanistic way. So he decided to go like from simple to more complex. So simple, just co-culture them in vitro, use tissue culture cells like HeLa cells, easy to manipulate, use human colonoids, which are intestinal, mini intestines, and also we use a model in bowels in vivo, so we can actually look at more deeply at those interactions, and I'll talk to that about later. So he wanted to look into mechanism. To look into mechanism, you have to be a bit of a reductionist. So what he did is he decided to use genetically tractable bacteria. EHEC is genetically tractable, and then on proteobacteria, he chose E. coli HS, which is a human commensal that's genetically tractable, and we know this is a true commensal. It went through thousands and thousands of human volunteers at the CDC in Maryland, never caused disease into anybody. Sequenced, you can genetically manipulate. Firmicutes, we chose Iphicales because Clostridium is a nightmare. And in the Bacteroides, we chose Beta, and they're all genetically tractable. Long story short, what I'm showing you here is EHEC in the presence of different members of this microbiota on human colonoids, okay? So those are really human intestine colonoids. Here, to confuse you, I have the bacteria in red and the cytoskeleton in green. So I know it's hard to see, so it's EHEC by itself, EHEC with Beta, EHEC with Iphicales, and EHEC with commensal E. coli. And you can see with electron microscopy that it's doing pedestals in this human colonoids. And here you can quantify this, so you don't have to, go blind trying to count pedestals in there. We made people in the lab count them. This is the amount of pedestals EHEC alone, it will make them. With Beta increases virulence of EHEC, I just told you they're friends, so you have more pedestals, more virulence expression. Same thing with the faecalis, and commensal E. coli and EHEC don't touch each other. So why? So Fernando decided to look at the metabolic landscape with the different combinations on human colonoids of these members of the microbiota and EHEC. And I'm not going to go through this because nobody deserves this. This is published. It's in supplemental material. If you want to go and go blind looking at metabolites and choose your favorite one, you're welcome to do it. It's not hard to find. It's free. But what I want you to see here is that whether it's EHEX by itself or with just faecalis or with all three of them, or just beta or commensal E. coli, the metabolic landscape in the colonoids changes quite a bit. So who is there is very important to have an idea of what kind of metabolites you're dealing with. And it's not just metabolites that it sees. It does see signaling molecules, okay? So it sees molecules that we usually refer to as autoinducers. This is the microbe's idea of hormonal communication, okay? This is called usually quorum sensing, and these molecules we call autoinducers. And it also senses hormones and neurotransmitters made by the hosts, which, it's the way whole cells communicate. So we can be one coordinated multicellular organism. And what my lab spends a lot of time in is in the intersection of these two, which we call interkingdom signaling, in which the bacteria are sensing at the same time bacterial signals, which is important. You have the microbiota in there, and host signals because you're also in the host and integrating those languages. And the signals that are sensing through the host, I'm going to spend my time today talking about neurotransmitters. And why neurotransmitters? When we first found this, I confess that I wasn't exactly jumping around super happy. Okay, but then we all remember, and now I think it's pretty accepted, that you have the gut-brain axis. Your intestine is super innervated. You have adrenergic neurons there. Most of your serotonin is made in the gut. You make norepinephrine there. So the bacteria in the gut are seeing neurotransmitters all the time. And this gut-brain axis is kind of a two-way street between host and microbe communication. There are strong correlations of microbial composition and microbial pathogenesis with host neurological diseases and behaviors. Members of the microbiota can not only modulate the levels of the neurotransmitters produced there, but also their activity in the gut lumen. And this exchange, as I will show you, does affect bacterial pathogenesis. And again, because there are so many drugs that deal with neurotransmitters, we wonder if we can manipulate that. So the neurotransmitter I'm going to be talking about today is norepinephrine. A lot of them are sensed by G-coupled PCR receptors in mammalian cells, GPCR. Bacteria tend not to use those. So they tend to use what we call two-component systems. I know the bacteriologists know there's 300 ways under the sun, but for those who are not, I'll just give a brief introduction. Usually the way this works is you have a histidine kinase. Most of them are in the membrane. They will sense a cue, okay? Phosphorylate themselves, start a phosphorylate cascade, phosphorylate what we call a response regulator, which is a transcription factor that go on to bind DNA and change gene expression. Okay, so that's the most normal signaling pathway that bacteria use. So the pathway I'm going to spend this whole talk talking about, and it starts with pathogenesis, is going to finish with something completely different here, is the one that senses epinephrine and norepinephrine. You can see they look the same. It's pretty much similar compound. And this signaling molecule produced by bacteria that we call autoinducer 3, AI3, because it was painful. It took us a couple of decades to get the structure. And then finally, Jason Crawford at Yale was able to get the structures with us. And those are a family of pyrazinones. This is the one that EHEC sees the best. The way this works is if it is a kinase, okay, QCC will have basal kinase activity. If you give in vitro separately, it's a biochemical assay, just AI3, which is the one signal coming from the microbiota, you get increased alpha-phosphorylation. It turns on the cascade. Same thing with epinephrine or norepinephrine. It works the same. And if you have the two of them together, they are agonistic to each other. And once this cascade goes, it virtually activates all of the virulence programs, not only in EHEC, but also other pathogens. Okay, what are epinephrine or norepinephrine? They are your fight or flight hormones or neurotransmitters. Norepinephrine, as I mentioned, is produced in the enteric nervous system by the enteric neurons, by the adrenergic neurons. Epinephrine is mostly produced in the central nervous system and the adrenal medulla, and it's dumped systemically. And remember, EHEC causes bloody diarrhea. So you have an infusion of blood in there at some point, which has also epinephrine in it. They control intestinal motility and secretion. They control the immune system and chloride and potassium secretion. So that's something that if you are a bacteria, you would like to read. So what is it controlling? All of the aspects of virulence in EHEC. So they will control expression of flagella, the lead, the type 3 secretion system, and also Shiga toxin, which is a toxin that's responsible for hemolytic uremic syndrome. So just to show you here, this is a lead gene, a flagella gene, a Shiga toxin gene. In black, it's expression in vitro just with AI3, and white AI3 plus epinephrine. And if you have a QCC mutant, which are the gray bars, you can see that it doesn't turn on because it's blind to the signals. So we spent a lot of time mapping the signaling cascades in vitro, okay? And because EHEC is a human pathogen, colonizes the colon, causes colitis and diarrhea, has this lead pathogen stylin that's absolutely important for colonization and virulence. And it makes these pedestals in the colon, which are the lesions that I showed you, but it won't do anything for a mouse, okay? Even though there are a lot of people out there who do not know that, okay? And even though Gary Frank and I wrote a whole thing on current microbiology reviews, a whole issue, apparently it might be 10 years old, so nobody read it. So if you want to use EHEC on a mouse, good for you. You're going to spend a lot of money with mice happy on the cage. So, and you cannot use humans, okay, because, well, you shouldn't do that. So with Citrobacter, so we use in the field Citrobacter rodentium. Think about this as EHEC for mice. It's a natural mouse pathogen, colonizes the colon, same place, causes colitis, has the same exact virulence genes, the Li, makes these lesions in the colon, and we use as a surrogate murine model for infection for EHEC because they have a very similar disease and they colonize the same places and have similar virulence strategies. So just to show that this is for real, this is a mouse that's susceptible to Citrobacter infection. And if you infect them with wild type Citrobacter in blue, they also come to death. So there's a survival curve. If you infect them with a QCC mutant, then QCC has no problems growing. It just cannot express the virulence genes. You can see that they are all surviving happy in the cages. And then came the experiment we wanted to do for a long time. Was a former postdoc in the lab, Chris Moreira, who has his own lab now at LSU, in collaboration with David Wenschenker at Emory, who is not a microbiologist, he's in the psychiatry department, okay, and he works with catecholamines. And David had these mice that do not make epinephrine or epinephrine. So this is the synthetic pathway. You have tyrosine, it goes all the way to dopamine. Then you have this enzyme here, dopamine beta hydroxylase, or called DBH, that will synthesize norepinephrine from dopamine. And then PNMT synthesizes epinephrine from norepinephrine. So the mice he had are knockouts for DBH, okay? They stop here. They have dopamine, but they don't make norepinephrine and epinephrine. This is homework in vitro. Those are several of those Lee virulence genes by themselves or in red in the presence of norepinephrine. You can see they're being expressed and turned on just like they are in EHEC, but in Citrobacter. And here, these are mice attached to the intestine of Citrobacter rodentium. And dark blue is wild-type mice that have everything in it. With wild-type Citrobacter, they colonize and attach to the epithelium. This is expression of the virulence genes, the lead genes in the intestine. They're being expressed at very high levels. Now, if you do the same experiments with the DBH knockout animals that don't make epinephrine or epinephrine, these signals are not there. So you have attenuation in the colonization and attachment to the intestine and pathology too, and also expression of the lead genes. So this is actually important and happening in vivo during infection. So I'll summarize this. EHEC comes in, it's seeing epinephrine or epinephrine, which is where we're going to focus now simply in the intestine through this QCC sensor to activate its virulence program. Okay, so this is a neurotransmitter. This will connect with the gut-brain axis. And, you know, there's been a lot of hype on the gut-brain axis because it has been linked with schizophrenia, autism, depression, all kinds of neurodegenerative diseases. But we're fun people, so we decided to look at drug addiction because there is a lot less in there, in that space. And this is a chronic disease. It's a compulsive, difficult to control, drug-seeking behavior, despite the fact that it's going to cause you harm, affect over 27 million people in the US, and costs north of $400 billion a year on not only health costs, but productivity, criminal justice important. And for a lot of the drug addiction cases, there is little therapeutic targets and not too many evidence-based treatments. The one we chose was cocaine, okay? Cocaine, the reason we chose that is because cocaine inhibits dopamine and norepinephrine reuptake. So usually in a regular firing situation, dopamine or epinephrine are thrown out there and then they are reuptaken and that stops, okay? Otherwise, I might die of a heart attack. Cocaine inhibits that. So that's why people on cocaine are all over the place and so excited. And this story starts with one of those serendipity situations in science. So I had this collaboration with David Van Schenker at Emory. And I told you he's in the psychiatry department. He works with catecholamines and addiction and not in microbiology at all. He has just 2 microbiology papers with us. And he was chairing a Gordon conference and decided to invite me to be a speaker. And I'm like, why me? And he's like, well, you're the variety speaker. So I go there to give a talk and everybody's looking very confused. Okay. So I finish the talk and then Santiago Cuesta comes to talk to me. And Santiago is like jumping up and down. If you know Santiago, he's not on cocaine, but it looks like he is. He does have his own lab now at Rutgers University, and he's a fantastic scientist, mentor in person. So Santiago comes to talk to me. He's like, I want to do a postdoc with you. And I'm like, why? I'm like, I'm a trained neuroscientist who work on addiction. I want to get to the gut-brain axis. And I'm like, I don't work on addiction. And he's like, Vanessa, you have a receptor in bacteria for norepinephrine and epinephrine. This is important for cocaine addiction and also amphetamine and methamphetamine addiction. I'm like, okay, let's try this. So, but then I looked at him, and since we're close to the World Cup, let's get some things straight first. Like, Santiago, I like you, but there is a little problem. He's from Argentina. I'm from Brazil. We do have a little bit of a soccer problem. So I had to talk him in some sense into him. And I'm like, okay, if we're going to work together, you're going to have to recognize one simple truth. The GOAT's Pelé. Numbers don't lie, 3 World Cups, Messi just won. Barely won. So, okay, we got that straightened out and started working together. So Santiago was like, okay, cocaine leads to addiction. because it increases the level of epinephrine or epinephrine, it might increase virulence. And we don't know what that would mean for addiction. Okay, so the first thing he did is he gave cocaine, and everything from now on is Citrobacter rodentium because we need to do this in mice, okay? So think about EHEC in mice, Citrobacter rodentium. So we first gave cocaine to Citrobacter rodentium. And unlike the guys in Wall Street, Citrobacter couldn't care less. And this was very good pure cocaine, by the way. So it didn't change growth, didn't change virulence expression, didn't do anything, you couldn't sense it. Like, okay, so it increases dopamine, it increases norepinephrine. Dopamine, Citrobacter rodentium also could not care. But we knew that it was sensing norepinephrine, that it was important, and it was turning on virulence. So it's like, okay, it has to be norepinephrine. So let's give cocaine to mice and see what happens. So these are the treatments. We basically have PBS with saline, PBS cocaine, infected mice with C. rodentium with saline or cocaine. And then we give them a lot of cocaine and then kill them and look at pathology. First, homework. It's been extremely well established that you have increased levels of norepinephrine in the brain if you give cocaine to mice. Okay, and we went ahead and just confirmed that. But we needed to be sure the same thing was happening in the colon of these animals, and it turned out that it was. It also increases the level of norepinephrine in the intestine. Okay, and for if there is any aficionados out there, we also measure dopamine and serotonin levels in the gut, and they don't change with cocaine. Okay. So then we did the experiment. So we looked at attachment and pathology in the intestine. So you can see here, mice with just Citrobacter get a lot of Citrobacter attached to the cecum. If you have cocaine plus Citrobacter, it increases. It's dependent on QCC. And this is dependent on the Lee because the mice who get cocaine and are affected with Citrobacter express more Lee genes here than the ones that didn't get cocaine, and this is again dependent on being able to see it. So the QCC mutant that can sense cannot do anything. This happens also at the level of inflammation markers. So these are several inflammation markers. Those guys are not infected in white and red, we don't care. The ones, this is early infection. So the ones in gray just got cocaine, they start to get inflamed. And then we gave them cocaine with C. rodentium, and you can see the inflammation is like to the roof. He also went ahead and looked at the non-inflamed ones just to make sure that cocaine itself wasn't priming, and it wasn't. There was no difference in the uninfected mice. So it was a combination of Citrobacter and cocaine. If you look at pathology, well, as far as I remember, PBS doesn't cause pathology in mice. Citrobacter rodentium does, and if it's Citrobacter plus cocaine, the pathology is up. Okay, and then we wanted to make sure what happens if we could block cocaine, but just in the intestine. So there is, so here is the levels of norepinephrine in the mice that are normal, and then we gave them saline with 6-OH-AGTA. This blocks production of norepinephrine in the gut, okay, not the brain. You can see that it's blocking norepinephrine here. And if you even give cocaine, it also blocks, it doesn't increase. And if you actually look what happens if you're blocking norepinephrine in the gut, either with or without cocaine, you do not see the bacteria attaching more, okay? So it is the cocaine in there. So, summary, cocaine increases the levels of norepinephrine in the gut. It's sensed by bacteria and Citrobacter through QCC, increases Citrobacter virulence and mirroring disease in mice. Okay, so this was kind of expected. That was a hypothesis. And I looked at Santiago, I was like, yeah, you know, cute, but it's boring. We kind of proved it, which is usually unusual, and which means it's probably boring. And he was like, yeah, well, let's see what happens to addiction. And again, showing my age. So how do you measure addiction? There are different types of ways to measure addiction in mice. So these are mice treated or not with cocaine. I labeled it for you, but I don't think I need to tell you which ones have cocaine on them, do I? They act just like human beings on cocaine. But as fun as it is to look at mice on cocaine running around in cages, you're a scientist, you have to measure and graph things. So what we did is we have these boxes that have lasers on them. So every time the mouse is running and cut a laser, it gets a reading that goes to the computer and maps and also plots it for you. So when we did that, we're like, okay, let's do our uninfected and infected mice with and without cocaine and see if it's affecting locomotion, which is one of the behaviors. Okay, because you saw how much they run. Okay, so who here thinks what's going to happen? So you saw that mice that are on PBS that got or not cocaine, the ones on cocaine run like crazy, right? With Citrobacter by itself, as far as we know, it doesn't affect locomotion. And we reasoned that the mice that got Citrobacter plus cocaine would probably respond to cocaine, but they were so sick that it was probably going to be an intermediary phenotype. That's a reasonable hypothesis. He comes to my office like, that's not what's happening. So this is mice that didn't get cocaine. We don't care. Here, PBS mice with cocaine, they're running around like crazy as they usually do. But if they get infected with Citrobacter and got cocaine, even though they're very, very sick, you just saw the markers, they're running more. They're responding more to the drug. They're like, okay, what we do now? And then like, okay, let's go with the hypothesis. Why is this happening? We're like, hey, everything in gut-brain axis turns out to be inflammation, right? You infect them with Citrobacter, they're inflamed. So it has to be something with inflammation. And you can see here, just to remind you, mice with just Citrobacter have a lot of pathology and inflammation. With cocaine and Citrobacter, they have more. So we're like, maybe that's what's causing the behaviors. So what we did is, let's take the Citrobacter out of there and just cause inflammation chemically using DSS and see what happens. DSS inflames mice, as it has been published 500 times under the sun. So this is no cocaine with and without DSS. This is cocaine with and without DSS. And you can see you no longer see that bump on behavior. So conclusion is it could not be inflammation. So the second one is like, changes in microbiota composition. So if you actually compare mice that are with PBS cocaine and Citrobacter cocaine, and you look at the microbiome of these mice in the intestine, what you can see here is that the real only difference is expansion of proteobacteria because they got Citrobacter. For the people who work with microbiome, we use Jackson mice, and we use them because they do not have proteobacteria. They will only have proteobacteria you give them, okay? In this case, it's Citrobacter. So Santiago looked at me, okay, we don't need the inflammation, but seems like we might need the Citrobacter. But does the Citrobacter need to be pathogenic if you don't need the inflammation? How about we just use a Citrobacter that's non-pathogenic, that's a mutant in the Lee, it doesn't type 3 secrete, and colonize the mice with that. And also let's just go wild and colonize with commensal Aedes coli, right? And see what happens. So first of all, just to show that in this case here, we have to pre-treat and deplete the microbiota to get them colonized, okay? And then we either give the fecal microbiota transplantation from mice from Jackson that do not have proteobacteria, or colonize them with the non-pathogenic Citrobacter or human or the commensal Aedes coli. And they all get colonized quite nicely. There's no pathology because none of those are going to cause pathology. And then we do the experiments. So in the bottom is no cocaine. Let's not bother with them. Here in black are the mice that got colonized with the SMT without proteobacteria. They're behaving like the PBS mice in response to cocaine. But if you colonize them now with either Citrobacter or commensal E. coli, you see the same bump in behavior, the same increase in cocaine behavior that to see if they had the pathogenic Citrobacter rodentum. So it is the fact that you have proteobacteria in there. So it seems to be the compositional shift. And then, you know, Santiago probably hates himself. He decided that he wanted to know why, to know the mechanisms. And I'm like, okay, kiddo. So he basically go, okay, let me look at the cerebral spinal fluid first of these animals. And this is a mouse, okay? Getting spinal cerebral fluid from mice, which you just get it from the brain, which is what's bathing the brain, is no joke. And then let's do metabolomics, because when you don't know what you were looking for, just do omics. So just showing you here that the metabolomics of mice that were colonized with either commensal E. coli or Citrobacter rodentum is exactly the same, which was super surprising. But it was very different from the ones who had no proteobacteria in there. I'm like, okay, great. So what's changing? I'm like, I'm pretty sure it's going to be like 500,000 metabolites, and God help us. The gods of science love this man. It was one pathway that lit up like Christmas tree. And it's all of the precursors in the pathway that leads to production of glycine. So here you can see is all of these metabolites on FMT. They're all normal, but they're all decreased in the mice that were either colonized with commensal E. coli or, sorry, Citrobacter rodentum or commensal E. coli. And I'm like, okay, what am I going to do with glycine? I'm not a neuroscientist, obviously. And he's like, glycine is really important. This is exciting. And I'm like, okay. He's like, Vanessa, glycine activates the N-methyl aspartate receptor, NMDAR, at the glutamatergic synapse. I'm like, awesome, English please. What does that mean? And he's like, this is associated with neuropsychiatric disorders like schizophrenia and substance drug abuse, especially psychoactive drugs like cocaine, amphetamine, and methamphetamine. And guess what proteobacteria do in the intestine, especially E. coli? They love to use glycine as a nitrogen source. So if you actually go and you look, we looked at the glycine levels directly, both in the CSF and in the intestine of these animals. So the ones with FMT, colonized with FMT, have normal levels, both in the brain and in the intestine. But the ones colonized with commensal E. coli or Citrobacter rodentum had depleted levels of glycine in there. And it's like, well, that can explain. And I'm like, it's okay. Is it really modulating the glutamatergic pathway? So he basically did RNA-seq. So the part of the brain that responds to cocaine is nuclear accumbens, here where these little balls are. And again, FMT have normal pathways, and the DSCAN and E. coli colonized mice, they had decreased glutamatergic synapses, which basically increases your addiction to cocaine. So, and respond to glycine as a ligand. So I'm like, okay, so if it is glycine, can we fix this? Can we give glycine back and get these animals to respond normally to cocaine? So what I'm showing you here is, so we have to pre-antibiotic treat them, and then we either infect them with FMT or C. rodentum or E. coli, commensal E. coli, and give PBS plus cocaine, PBS plus saline, and cocaine with glycine. Okay, and let's see what happens. And this is what happens. I know this is easy. So these guys here in the bottom didn't get cocaine. Here on the solid lines, the ones in black, it's FMT. This is locomotion again. FMT with no commensal E. coli. They respond normally like the PBS mice with cocaine. If you get them colonized with Citrobacter or commensal E. coli, you have the increase in the phenotype like we've seen before. But now if you colonize them either with commensal E. coli or Citrobacter, with cocaine, but give glycine, they go back to as if they didn't were colonized, like the FMT mice. So you can correct the phenotype. And then we're like, okay, the other thing, you have all of these metabolites here that lead to the synthesis of glycine, okay, that are all depleted. And bacteria use glycine, but they don't use sarcosine. So you deplete glycine, you also deplete sarcosine because your cells are trying to make glycine. And the fun thing about sarcosine is this. So we could do the same correction with glycine. So what you're seeing, sorry, with sarcosine. So here is the guys who have FMT, and here and cocaine. Here is with Citrobacter or E. coli, cocaine, or glycine. You know, it brings it back. And why is glycine fun? And this is when you can make fun of your kids. So sarcosine is a food supplement that athletes, especially gym bros, like to use to increase muscle. And you can go to the GNC and buy them. And as you can see, I have my gym bro kid. Oh, he so deserves that. Okay, who I spend a lot of money on meat for, who uses this. So you can actually do the same thing with a supplement. So glycine reduction increases cocaine responses, but we needed to see whether the proteobacteria was indeed doing this. So what he did is he made a E. coli HS mutant that does not use glycine. Okay, it's a CK mutant. Uses other sources of nitrogen, but no glycine. And as you can see, this is glycine levels in the intestine and the brain. Normal if it's FMT, decreased if it's commensal E. coli wild type. But if you have the commensal E. coli that can't use glycine, it does not deplete. Okay? And this is just colonization in the gut. We're not doing anything funny. And if you look at locomotion, no cocaine, we know what happens. FMT, you have normal locomotion. You have the wild type commensal E. coli, you have the increased phenotype, okay, because glycine is going down. But if you colonize them with the CK mutant, it won't use glycine, so you don't see the bump in the phenotype. So, and then he comes to me and is like, yeah, this is great, but you know, we're gonna be creamed if we just go with one behavior. And I'm like, what other addiction behaviors, man? It's like drug-seeking behaviors. So you do that by using conditionally placed preference, which is a drug-seeking behavior. You have these two boxes here that have a door between them, and they have different tactile and visual cues to them. And if you put the mouse here, they will spend the same amount of time in both chambers. Okay, but then what you do is you just start giving cocaine for them in one of the chambers. Then you take the cocaine away and see if they develop a preference for where they get cocaine. Okay, just a CPP score. Long story short. So this is pre-tests, they don't have a preference. Post-tests, it would be a preference if it's different. If they have FMT colonization, they don't develop a preference. You can see they're non-significant. If you colonize them with commensal E. coli or Citrobacter rodentum, they develop a preference. They start seeking for the drug. And if you colonize them with the CK mutant, the one that does not use glycine, you do not develop the preference. And then because Santiago hates himself, he decided to do self-administration of cocaine in mice. So what you do is you put a catheter in the aorta of the mouse. I'm not joking, okay? And you have to do all kinds of weird things to make sure a biofilm doesn't form in there. You also have to make sure you don't kill them because, you know, you're putting the catheter there, but you're also inoculating them orally with all kinds of things. And not only that, you have to first train them to place a lever with sugar. And mice are dumb. 50% of them are not trained, do not train. Then there's all of the ones that you kill trying to put in the catheter, or that you dislocate the catheter when you're trying to infect them. But long story short, he's persistent. So what you're seeing here is the score. They're both pretty much the same thing, just different ways of looking at it on how much they press the lever. So if they're colonized with wild type E. coli, they like cocaine. We established that they press that lever a lot. If they are colonized with the CK mutant, they do not. And then he wanted to look at relapse. So what you do in this case is you give, this is again, self-administration. You give cocaine to the mice, you take the cocaine away, you extinguish it for several days, and then you reinstate the drug and see and measure how much they press the lever. Okay, and remember, you did the colonization here, and then by that time, okay, we stopped doing the colonization with the different protobacteria in there. The microbiota starts reestablishing, but something changes, and they are still pressing the lever if they had been previously colonized with commensal E. coli, way more than the one that does not use glycine. So it also has something to do with relapse. To finalize then, and this is very preliminary, and Santiago is the person who's going to be doing that in his lab, he wanted to see what happens with other members of the microbiota. So this is just locomotion. So is everybody bad for you or not? So we know that if they don't have, if it's just F and T, the locomotion is normal. You know that with Clostridium, by the way, is as bad as E. coli. It increases the drug behavior. But beta is nice. So I hope we all get a lot of beta because it actually decreases the behavior. So the composition may actually change. So cocaine exposure by increasing norepinephrine levels in the gut will lead to a protobacterial bloom. Actually, if you get mice that have protobacteria there and give cocaine, increases the levels of E. coli. And humans who use cocaine do have more E. coli in their microbiota. This protobacteria use glycine as a nitrogen, depleted in the gut, depleted in the brain, that alters cocaine plasticity in the neck and increase addiction behaviors. You can fix that with systemic glycine or colonizing them with an E. coli that does not use glycine. And so, and other microbacterial members seem to differentially sensitize the host towards drug utilization. So to summarize here, because we had to do this figures nowadays by biorender. So you give cocaine, more norepinephrine sends to QCC, expansion of protobacteria. You ended up having depletion of glycine in the intestine and in the brain that messes up with the glutamatergic synapse and increases addiction-like behaviors. So in summary, I tried to talk about everyone while I was talking about them, but all of the cocaine stuff has been done by Santiago Cuesta. David Van Schenker at Emory helped us with mice that don't make epinephrine or epinephrine. Sid Courage, who is now at the University of Quebec, and David Self at UT Southwestern helped us with the cocaine behaviors, because needless to say, I'm not a neuroscientist. Jason Crawford helped us to get the structures of AI3, and John Leon to get the Citrobacter models going. And that's it. So.
Time for a couple questions, and so.
Yes.
So you mentioned that Clostridium is as bad as protobacteria.
Yes.
And beta is.
Seems to help.
When you do have a mix of those two, we just know microbiota compositions. What's the behavior?
Since I was doing those things, I'm not touching this anymore because I'm not competing with my farmer postdoc. So wait, wait and see what he finds out. Yeah.
Are any of the effector expression, like, does it seem like cocaine induces a specific like array of effectors, or does it go out through the type 3 secretion system, or is it still kind of like the same arsenal of effector proteins?
It's so it's all of the arsenals, because a lot of so you have inside of the Li, you have about 5 effectors that are type 2 secreted, and then you have several effectors, because ZHEC has about 51, Citrobacter 22 that are in other pathogens, the islands, but they're all controlled by the same transcription factor that controls the Li. So if you hit there, you hit all of them.
So. Go back to great model. I mean, what is the trend?
It's the pain of my existence. If I can ever convince somebody to do that, I will. So, the parts of the type 2 secretion system, which is like the translocon, which is SP, B, and D, which are the part that touch the cell first, those proteins are very variable between Citrobacter and EHEC. So I think it's a receptor issue, kind of like what happens with Listeria. And nobody knows what the receptor for the type 3 secretion system really is.
If you exchange, can't you?
That's what I've been trying to convince somebody to do for a long time.
Okay.
Okay, and other than put a gun on somebody's head, I don't know how I'm going to convince somebody to do that. I think I finally convinced one student. Okay, yeah, I agree. Yeah.
So, guess like this is a really big system, and you showed like it also has like similar effect to Clostridium. But how common is this sensing system in Proteobacteria? Or another question along that way is like, is that how EHEC senses the colon, like the norepinephrine system or the levels?
So QCC is actually very conserved in gamma proteobacteria throughout. Okay, and in several of these cases, it does always sense epinephrine or epinephrine. Okay, so it's very conserved, and there is a lot of norepinephrine there in the colon, and because it's made there, it's in the micromolar range, which is higher than most of these receptors even need. So in Clostridium, we have no idea how this is actually happening. Okay, and because Clostridium does not have a homolog of QCC, so it could be a very different type of system that you're dealing with. And yeah, it's his job to do that now.
Also, I mean, the Proteobacteria colonizes differently, right? Salmonella colonizes the ileum, and then EHEC goes to the colon. So is there like something else that EHEC senses, or
So with salmonella, so EHEC will sense a bunch of other things, as you know, right? So it will sense epinephrine, norepinephrine, endocannabinoids, serotonin, adenine, what else it senses? Oh, folate. So it senses a bunch of things, and not all of those will be very well conserved. For example, with salmonella. The complication with norepinephrine with salmonella is not just the location, okay? Because you have more, at least old literature with human cadavers, you have more norepinephrine in the colon than you do in the small intestine. You still have, but it's way more in the colon. And it's the opposite, for example, for things like serotonin. So, but I mentioned briefly that norepinephrine, epinephrine control the immune system. And we do have a paper with the DBH minus mice and salmonella. So we can see the control of the type 2 secretion systems, especially the second one, okay, the systemic one. But it's a problem because there's also problems, immunological problems in controlling intracellular bacteria if you start messing with the levels of epinephrine or epinephrine in the host. Because Citrobacter and EHEC are not intracellular, it's easier to see the phenotypes and it's less convoluted.
question? In the cocaine mice, is your Citrobacter restricted to the gut, or not restricted to the gut, but does it also go to the brain?
No, it doesn't. Just stay in the gut. Yeah, it doesn't cross. Citrobacter will cross and go to some systemic sites if you're using very, very, very young mice, which is the work from Gabriel Nunes and Gustavo Caballero Flores. So they have what they call baby model. I think that's what they're calling it. So if they're using the baby model, the baby mice have a problem controlling Citrobacter in the gut. So that one will cross, and they know, I think they looked at the liver and spleen, obviously, right? Pyrus patches. I'm not sure they looked in the brain. I think it's unlikely to go to the brain because I don't think it can cross the gut-brain barrier.
Just wondering about the cocaine. If it doesn't.
Mhm. No, we know the cocaine, what's happening is the cocaine, so the whole thing is going systemically through the bloodstream. So you change here in the gut, and it changes the amount of the glycine that goes through the bloodstream that reach the brain. But what we can see is systemic gut, bloodstream, brain. Yes, it is. So in Texas, you only had to fill the DEA forms, which were not that awful. In Wisconsin, you have to do the Wisconsin forms first. So I've been waiting for about 6 months for the one person in the office to go through the Wisconsin one, so I can then go to the DEA one. I don't know why, but yeah. And then once you get it, you have to have, it's like having catecholamine in your lab. It's a Schedule II drug.
All doses for mindset.
It is, but it's very pure cocaine too. It's the kind of thing that, and I couldn't bring my cocaine from Texas to Wisconsin.
All right, I think we should wrap up. Let's get one more.
Mhm.
She'll send you.
Oh, okay, sure. Yeah, yeah.
You said there are bunches of.
They're like the same. You can look it up.
Einstein, your.
Which part?
That's that.
The cocaine, I'm not sure. With, if Methanol was sure with someone else decided on that.
Yeah, like, yeah, that or, dude, what, one looks like fully.
Yeah.
She gets some safe.
I guess.
She gets some safe.
Yeah, let me.
I guess.
No, 'cause